Are you struggling with fatigue or brain fog? The MTHFR gene might be the reason. Nearly 50% of all people have a variant of this gene, and it affects methylation, a process your body depends on for energy, brain function, hormones, liver detox processes, and even insulin resistance. The good news is you can support it with the right foods, careful use of B vitamins, and simple lifestyle shifts. This gene touches everything from mood to metabolism. This is lecture 107 of the Metabolic Classroom. Looking to improve your own metabolic health? Visit insuliniq.com for courses, coaching, consultations, and a 10-day free community membership trial. To dive deep into the science behind metabolic health, become an insider at benbikman.com, where you'll enjoy my exclusive content, ad-free podcasts, live stream Q&A access, and more. Welcome to the Metabolic Classroom. I'm Benjamin Bikman, a biomedical scientist and professor of cell biology. Today, we are discussing the MTHFR gene, a cornerstone of cell biology, and its regulation. This gene drives a process called methylation, which is critical for homocysteine metabolism, DNA synthesis, gene expression, and, as you will learn, much more. When MTHFR function is impaired, it can lead to things like oxidative stress and myriad metabolic challenges, including some substantial effects on mitochondrial function. In this lecture, we'll unpack the MTHFR gene's primary roles, explore its metabolic implications, and even some evidence-based strategies to support your health. My goal, of course, is to empower you with knowledge so that you can optimize your metabolic health. Now, let's begin. In fact, let's begin with the core functions of the MTHFR gene. MTHFR is short for methylene tetrahydrofolate reductase. Now you're hearing the A's at the end, which means it's an enzyme. So this gene produces an enzyme that plays a crucial role in methylation. Methylation is a biochemical process where small chemical tags called methyl groups are added to molecules. This process regulates countless cellular activities, including DNA repair, gene expression, liver detoxification, and amino acid metabolism. In fact, methylation occurs billions of times each day in every cell of your body. Specifically, the MTHFR enzyme helps convert one form of folate, called 5,10-methylene tetrahydrofolate, into another active form known as 5-methyl tetrahydrofolate, or 5-MTHF. This active folate is essential because it donates methyl groups to homocysteine, converting it into methionine, which is an amino acid that's vital for protein synthesis and countless metabolic pathways. However, not all MTHFR genes function equally. Some people carry genetic variants, the most common being the C677T polymorphism. And this can reduce the enzyme activity by 20 to 70%. And importantly, this is thought to occur in up to 30 to 50% of people. So it's remarkably common. In another common variant is the A1298C version, which also affects the enzyme function. Now, as I noted, both of these are quite common, and about half of all people are going to have one of these. So half of all people are going to have a version of the MTHFR gene that will impact the methylation efficiency. This matters because when methylation is impaired, homocysteine can accumulate in the blood, and it is a known risk factor for inflammation and cardiovascular disease. These MTHFR variants also reduce the production of 5-MTHF. Of course, that's not surprising. That's the product of that enzyme that's coming from that gene. The result of this is that you can have a disruption in the metabolism of critical B vitamins like B12 and B6, which are both essential players in methylation. Altogether and ultimately, this creates a cascade effect. less efficient methylation, disrupted nutrient metabolism, impaired support for things like DNA synthesis and repair, especially in cells that have a very rapid turnover, like red blood cells and even white blood cells. Now, beyond the things I just mentioned, methylation also regulates gene expression, essentially determining which metabolic pathways are turned on or off. These foundational roles, methylation, homocysteine regulation, etc., illustrate just how central MTHFR is to overall cellular biology and even metabolic health. Now, let's dive into the metabolic aspect a little more. One primary problem is oxidative stress. It is a primary outcome. But I also want to touch on mitochondrial function and, of course, even some insulin resistance. Oxidative stress arises when MTHFR variants elevate homocysteine, increasing reactive oxygen species, ROS. This oxidative stress damages cells, disrupts vascular health, that's a particularly susceptible part of the body, and drives inflammation, creating a cascade of metabolic dysfunction. Mitochondrial function is also affected. Methylation via 5-MTHF supports mitochondrial DNA maintenance and nucleotide synthesis, especially the famous ATP. ATP fits in that description, and it is affected by all of this. Impaired folate metabolism reduces mitochondrial function, lowering ATP and potentially contributing to increased susceptibility to fatigue. Now, that's limited and somewhat weak evidence in humans, but it appears to be fairly consistent, this increased susceptibility to just being slightly more fatigued. If you have a hard time creating ATP, you're going to have a hard time getting any type of work done. MTHFR variants further reduce a molecule that is just abbreviated as SAMe or SAMe. Sometimes if you see it abbreviated, and you probably have, it's capital letters SAM and then lowercase e. But this is a methylation product that supports mitochondrial biogenesis, so the synthesis of new mitochondria. Elevated ROS from homocysteine damages mitochondrial membranes, linking oxidative stress to energy metabolism. Now, insulin resistance, which of course is a focus of my research, is also associated with MTHFR variants. Though this is observational, but I'm going to mention in a moment that there is a mechanism that supports this. But elevated homocysteine increases ROS, which impairs insulin signaling, and that can then affect GLUT4 translocation. Now, this is a whole area of research looking at the degree or the mechanism whereby oxidative stress can impact the insulin signaling cascade. Some people will say that it is affecting the IRS, the insulin receptor substrate, which is right at the very beginning of the insulin cascade. Others will implicate that it influences AKT phosphorylation, which is a further downstream section of the insulin cascade. But suffice it to say, if you have a variant of MTHFR and the elevated oxidative stress, you are going to impact the insulin signaling cascade. And as insulin signaling is disrupted, one obvious consequence of that is going to be elevated blood glucose. Now, I want to go a little further into the cell before we revisit insulin resistance, because the story, if you will, of impaired methylation doesn't end with homocysteine and B vitamins. Methylation reaches into the very core of how the genes function. It is, in fact, an important part of cell biology. Methylation is one of the key tools that our body uses to control gene expression. When methylation is working properly, it helps keep the right genes turned on and the wrong ones in that moment turned off. But when methylation is disrupted, that balance can break down. And this absolutely has very profound implications for long-term health. For instance, improper methylation can lead to epigenetic dysregulation. Like I just mentioned, where you have genes that should be silenced are accidentally activated, and protective genes in that moment are shut off. This, of course, has implications for things like cancer, but it even has implications for the brain. Your body relies on methylation to produce key neurotransmitters like dopamine and serotonin. When the process falters, it can contribute to mood disorders like depression and anxiety, as well as impair cognitive function and memory. Even further, hormone balance is another victim. Methylation plays a critical role in the clearance of estrogens and other steroid hormones. If that process is slowed, estrogen metabolites can build up, contributing to hormone-sensitive cancers, menstrual irregularities, and even symptoms of estrogen dominance like fatigue, weight gain, or irritability. Finally, methylation governs genes involved in the immune response and inflammation. When that regulation is lost, it can promote chronic low-grade inflammation, which is a very well-known driver of insulin resistance. And in fact, on a personal note, the very topic that got me interested in metabolic research in the first place as a young master's student so many years ago. So while it may seem like a small enzyme, MTHFR's role in methylation touches nearly every corner of human health, from mood and memory to metabolism and immunity. Now, let's turn our attention to how MTHFR and methylation influence nutrient metabolism, particularly where there's most to discuss fat metabolism, which is, of course, very relevant to overall metabolic health. But it's not where you think. It's not within the fat tissue. When MTHFR activity is compromised and the methylation process slows down, the effects ripple, but it's largely focusing on the liver. When methylation is impaired and that level of the SAMe fall, the body's ability to oxidize or burn fatty acids can become less, capable. In a healthy system, the fats are broken down in the mitochondria and used for energy. And the liver is a good spot to focus on because the liver can do a lot with every single nutrient. That's why I refer to the liver as the soccer mom of nutrient metabolism. It just is involved in everything that's going on, whether it's lactate or ketones or amino acids or proteins, fats, and glucose. The liver is the one single tissue that can handle all of them. But in this particular context, the shift can become quite apparent in the liver, where studies have shown that when MTHFR activity is even moderately reduced, like what you do see with these genetic variants, it can lead to an accumulation of fat in the liver cells. And the buildup of triglycerides and cholesterol esters and phospholipids, all of which can accumulate when the liver isn't burning fat well, like with these MTHFR variants, it absolutely sets the stage for fatty liver disease. This is all the more relevant if the MTHFR variant is combined with a high insulin diet. Now, of course, some of you might be rolling your eyes and you're thinking, Ben, surely you don't have to talk about insulin all the time. Well, maybe I don't, but why would I want to when I can so easily invoke this petite peptide and its relevance to so many processes? Why do I say the high insulin diet compounds all of this? Because if the MTHFR variant and its effects on methylation are influencing the liver's ability to burn fat, you don't want to gum up that process or block that process any more than it's already blocked up. And when insulin is elevated, it inhibits fatty acid oxidation. In fact, essentially every enzyme that's involved in the burning of fat for energy gets turned off when insulin is turned on, if you will. That's why I mention the high insulin diet. That's going to compound the propensity of the liver to be storing fat rather than burning it. Now, on top of that, elevated homocysteine, which is the, of course, obvious hallmark of impaired methylation based on the role of the MTHFR gene and enzyme in the first place. It can increase oxidative stress. And a topic that I've spoken to previously is that when oxidative stress is up, one consequence of this is going to be the peroxidation of lipids. Now, earlier I mentioned oxidation. I try to be quite deliberate with my use of these words. When I say oxidation, I mean burning a fat for energy. Now I'm saying peroxidation, which is when you are basically creating you're taking the reactive oxygen species and you are using that to create a reactive lipid. So this is where you start to have these lipid peroxides, which are going to further contribute to not only liver problems, but problems everywhere throughout the body. And this is much of the reason why the omega-6 polyunsaturated fat linoleic acid, which is enriched in refined seed oils, is so problematic because it undergoes peroxidation so readily. Well, suffice it to say, an MTHFR variant may make that even more likely as linoleic acid is to already undergo peroxidation and become a very reactive, damaging version of itself. If you have an MTHFR variant or MTHFR is not working well, you may be amplifying that concern. Now, I've alluded to this, but this is a connection that often goes overlooked. And of course, knowing me, you're not surprised that I wouldn't overlook this. And that is how elevated homocysteine can actually interfere with insulin signaling itself. Now, earlier I had mentioned the somewhat indirect effect through oxidative stress, but homocysteine can chemically modify the insulin receptor itself. Now, as a reminder, the insulin receptor is essentially the small docking station, or I often refer to it as a door, that is designed specifically for insulin to come and knock on. In a process called cysteine homocysteinylation, homocysteine will bind to and alter the structure of the insulin receptor precursor. Now, you've not ever heard me describe that, but the insulin receptor precursor is the early form of the receptor that is made within the cell. So before the insulin receptor comes and binds into or inserts itself, that's the best word here. Before the insulin receptor, which was formed within the cell itself, comes and inserts itself into the membrane of the cell, thereby acting as a dock for insulin as it's coming outside the cell, it exists within the cell in a precursor form. Well, homocysteine will compromise that maturation process. So as homocysteine accumulates, it can prevent the receptor from maturing properly. And so rather than maturing and coming to the cell membrane and inserting itself into the cell membrane to realize its role in all of its glory, it stays immature. It's locked in the cell. It's essentially a failure to launch. It's a kid who doesn't move out and is now living in its parents' basement. But it's not the kid's fault. It's not the insulin receptor's fault in this case. It is extenuating and external circumstances, namely the elevated homocysteine. So the result of this, of course, at the level of the cell, is that the cell cannot respond adequately to insulin's signal. So insulin is flowing through the blood, coming to the cell, looking for a door on which it can knock, but there's nothing for it to knock on. So the cell's not responding because the insulin receptors aren't able to get into the cell membrane to do their job. Again, the receptor becomes damaged, if you will, at this very molecular level within the cell. But all of this, as I mentioned earlier, this mechanism here does at least partly explain why we see such strong and consistent correlations between elevated homocysteine and insulin resistance across so many human studies. Higher homocysteine levels are frequently associated with higher fasting insulin, elevated HOMA scores, and indeed an increased risk of type 2 diabetes. So as a result, homocysteine is not just a biomarker. It is, in fact, an active mechanism driving some degree of metabolic dysfunction. All right, now, in the midst of all of this science, there are, I believe, some practical takeaways. So let's translate some of these ideas into some action. And this is, of course, particularly relevant for those who have or suspect they have one of these less effective MTHFR variants. And I'd noted the two that are the most common, and again, surprisingly common, potentially affecting up to, I think the ranges are often cited as about 30 to 50%. First, I think it is helpful to get some clarity through testing. A simple blood panel measuring things like homocysteine, folate, and vitamin B12 can give you insight into your current methylation status. And if you go deeper, of course, genetic testing can confirm whether you carry common MTHFR variants, like the C677T or the A1298C versions. But then next, it is very prudent to be smart about how you get your folate. Animal sources are incredibly relevant and even efficient here. Just 3 ounces, for example, of chicken liver will provide about 500 micrograms of folate. And then beef liver will offer about half of that. But interestingly, both of them are coming or providing the highly bioavailable 5-MTHF form that the body can readily use. So again, it's giving you the already reduced form or active form that if you have an MTHFR variant, that's what you're having a hard time making. But if you're eating these liver sources, you're getting that source. Now, plant sources, when you talk about folate, may still be valuable, but there's a little more nuance there. People will often say that leafy greens are good. Well, only if they've been cooked, of course. The ability to get this is in an uncooked form, like if you're getting your spinach or your kale, which will give a small degree, maybe about 100 micrograms compared to the potentially 500 micrograms. If it's uncooked, you're getting even less than that. If it's cooked, you're going to get about the amount I mentioned, about 100 micrograms. But it's not that simple. The folate in these greens comes in what's called a polyglutamyl form, which needs to be further enzymatically converted before there can be even any absorption. So cooking the greens, like steaming or sautéing, can help make the folate a little more available by helping break down the cell wall that's locking it in. But even still, you have further processing required. So I do absolutely think unapologetically that it's more prudent to try to get it from animal sources if you're trying to obtain it in your diet. Now, further, beyond folate, you could act to support methylation machinery, the enzymatic machinery with foods that are rich in other nutrients, like trying to get more B12. And of course, B12 is only going to be coming from animal sources, like, well, any eggs and fish. But you could also be looking for more B6 from nuts, for example, and then even choline. Choline from eggs is known to be a helpful thing here as well. Now, finally, if your blood tests actually do show deficiencies or if you have some symptoms, you could consider some targeted or smart supplementation. In particular, using methylated forms of B vitamins like L5-MTHF for folate, methylcobalamin for B12, and P5P for vitamin B6. But of course, none of this is medical advice. I'm just your friendly neighborhood scientist. You absolutely ought to consult with your healthcare provider just to make sure you're tailoring any dosing and proper supplements. This concludes today's lecture in the Metabolic Classroom. We've explored the MTHFR gene's canonical roles in methylation and homocysteine regulation, its metabolic implications like oxidative stress, and even, I hope, some practical steps to optimize your health. Of course, I hope more than anything else that you appreciate the role that it has in insulin resistance, the primary pathology that I believe everyone ought to be very intimately worried and thinking about. Thanks for joining me. Remember, more knowledge, better health. Looking to improve your own metabolic health? Visit insuliniq.com for courses, coaching, consultations, and a 10-day free community membership trial. To dive deep into the science behind metabolic health, become an insider at benbikman.com, where you'll enjoy my exclusive content, ad-free podcasts, live stream Q&A access, and more.